Good afternoon to everyone. First, I'd like to thank the ISMB and ECCB committee for accepting my abstract and the SFBI for their support. I'm Farhat, postdoc at the National Museum of Natural History, and I will now present to you the work I have carried out about whole genome duplication and gene evolution in a hyperdiverse group of gastropods, which is part of the hyperdiverse project led by Nicolas Pilon. First of all, what are neogastropods? They are a group of marine predators with 15,000 described species, representing 22% of gastropod diversity. In addition to their large number and diversity, they have a range of feeding strategies, such as envenomation by injecting neurotoxins, asphyxia by injecting anesthetics, or vampirism as they feed on their prey's blood by injecting anticoagulants. In the Cynogastropoda, the subclass containing the neogastropods, only 14 genomes have been sequenced and published, with highly heterogeneous genome assembly qualities. 8 of these genomes belong to neogastropods, our group of interest, but with a majority from the Conoidea superfamily, known for secreting conopeptides and of pharmaceutical interest. Unfortunately, currently, there is only one high-quality genome available for this superfamily, as it has been assembled at the chromosome level, here Canis ventricosus. Apart from Conoidea, 4 genomes of ampullarids are the relatively good quality, particularly Pomacea, the freshwater apple snail, which has been assembled at chromosomal level. What was found about Canis ventricosus is that it was subjected to a whole genome duplication, which is an event where each chromosome of a genome is duplicated. Let's come back a little bit before this discovery. A 2011 article by Hallinan and co-workers detected a possible whole genome duplication in the neogastropods and a portion of the Lithoronimorpha, as you can see on the tree here on the left. This prediction is based on the number of chromosomes and genome size of these species. Then Carlos Blas' article, which described Canis ventricosus, validated an event of whole genome duplication, and their team successfully assembled the genome at a chromosome level. Here we can see that for each of the 14 chromosomes of Pomacea, they have homologies to at least 2 chromosomes of Canis ventricosus. Our main interrogation here is why this group of neogastropods is highly hyperdiversified and what molecular processes could explain it, especially if there is a connection with whole genome duplication. To begin working in this direction, we selected 2 species. I have included here one of the latest phylogeny made by Alexander Filozov to have an overview of the distance between the species we have selected. Monoplex corrugatus is a sister group of the rest of the neogastropods, with Conoidea, which is also a marine predator that we have collected from Corsica, and Stramonitiae mastoma, which is a myriad collected from Italy. As you can see, there is a representative of superfamilies whose genome have not been sequenced yet and are divergent from each other and Conoidea. So briefly, for the method, PacBio was used as a sequencer with the addition of the OmniSeq technology developed by Dovetail, which allows the sequencing of contact points in the genome using Illumina sequencers. Combined, we were able to reconstruct the chromosome of our 2 genomes of interest. Here I'm showing the contact matrix generated that helped in reconstructing the 35 chromosomes of monoplex corrugatus. The same was done for Stramonitiae mastoma. With the help of RNA sequencing, we also have conducted the detection of repeats and gene annotation. So here we have the statistics of these genomes, and we can notice that for Stramonita, it showed a little smaller genome size of 2.2 gigabase compared to Monoplex and Canis. While comparing to Pomacea, the freshwater snail I presented earlier, which is 6 to 8 times smaller than the 3 others. It also has more than half number of chromosomes, while the 3 neogastropods have 35 chromosomes each. Finally, the number of predicted genes is similar between the 3 neogastropods and is the double of the number of genes predicted in Pomacea. This made us wondering about whether duplication events happen in our selected organisms. So we have computed the centenary analysis for monoplex and Strammonita. And as you can see on the left figure, blocks correspond to chromosomes and red and blue lines represent centenic blocks between them. We are showing here the comparison between the 4 chromosome level genomes. So for 1 chromosome of Pomacea, we observe 2 to 4 chromosomes in the neogastropods, similar to what was found for Conus ventricosus. An interesting fact is that if you look at the centenary within each of these species, there is very, very few centenic blocks that we can detect in the genome of each of these organisms. In addition to centenary, another way to verify whole genome duplication is by looking for the Hox genes, which are known to have a highly conserved cluster in a specific chromosome, and timing this event by calculating the distribution of KS. On the left, you can see a representation of parts of the chromosomes where we identified Hox genes in different organisms. In the 3 neogastropods, there is clearly a duplication of the cluster, one copy having all the Hox genes and another one with some of them missing. While in Pomacea and, for example, Pecten maximus, which is a scale-up, there is only one cluster found on 1 chromosome. We also calculated KS for first the paralogous genes within each species of neogastropod, where you can distinguish 3 peaks. The first high peak followed by 2 very small peaks. The same analysis was done on orthologous genes between the neogastropods, where we can better distinguish the 2 following peaks. And finally, by comparing orthologous genes between Pomacea and the other 3 species, we observe a clear older peak that corresponds to the divergence between Pomacea and the neogastropods. Therefore, the second peak here in the middle likely corresponds to the time of whole genome duplication event. So to conclude on this part, the higher number of chromosomes, the larger genome size, centenic blocks, the duplication of Hox genes cluster, and the KS peaks clearly confirm a whole genome duplication in neogastropods. So now, one of the interests of this project is also to discover new toxins in this species. Therefore, we specifically manually annotated genes related to toxins and found 166 and 130 such genes in our 2 species respectively. From these, we were able to detect 12 groups of genes present in all 4 species and within centenic blocks. Here on the left, there is an example of one of these groups. I'm not going to detail this figure, but you can see in a red line the putative toxins that are all orthologs and found in different parts of the chromosomes of the genomes of interest that were found in centenic regions. On the right, you can also see a sequence alignment between all members of this group, which shows similarities between the sequences. So then we were wondering which kind of evolution after the whole genome duplication were the gene resulting from. As you can know from an ancestral gene function, after a duplication event, different possibilities can happen, like non-functionalization, meaning that the copy is eliminated and only one copy remains, which is the most common fate, or conservation of both functions, which would have an impact on the expression of this gene. Or new functionalization, where the copy would diverge enough to imply a new function, or a subfunctionalization, where both copies would work together to maintain the ancestral function, and finally, specialization. So here we had RNA-seq triplicates from 2 tissues of Strammonita hemastoma. The host sprayed them, which is a chemosensory organ, and the salivary glands known to produce venom. So we performed differential gene expression and found 6,000 genes which were differentially expressed. From these, we looked for their copies in the genome and if they were homologs, meaning orthologs found on the homologous gene, chromosome. The total number of homologs were only of 260 and 370 for the up and down regulated genes respectively. For most of the copies having differential expression, they had the same pattern of expression. So here in red are the toxin we found differentially expressed. Of course, we can't conclude with such little data, but it gives an idea about the pattern of expression of homologous genes. To be able to determine if the toxins were resulting from sub or neo-functionalization, we would need more expression data within different tissues of the organism and comparable ancestral organism. So here, thanks for your attention.